Hello, my name is Tyler Duquette, and I'm here to talk to you about synthetic biology, what it is, and why you should care. Alright, to start, synthetic biology is the manipulation of living cells or synthesis of new cells from synthetic materials to achieve a desired outcome. And what a desired outcome means is really scientists want to manipulate an organism to achieve a goal, whether that goal is to produce a molecule, to produce a chemical, or that goal is to be used as a diagnostic. It's a very, very broad term because there are many different techniques, industries, and technologies that it encompasses. Um, Synthetic biology is an up and coming technology, and the reason I say that is because although it has existed in the past few decades, recent advancements in technology like CRISPR-Cas9 and in machine learning like AI make the potential growth in this field extremely high. There are well-documented genomes of many organisms, and with machine learning, the manipulation of those genes can be very, very easy. Okay, the first major application of synthetic biology are biosensors. A biosensor is a specialized cell that can basically sense the presence of a targeted molecule in the environment. The earliest examples of these were called hybrid devices. These hybrid devices were physically mounted onto a larger detector and they basically sent a signal that let the detector know that there was something in the environment that they were targeting. They were very limited use and they didn't have widespread application, but this then evolved into RNA-based biosensing in living cells. And RNA in nature plays a big role in reactions and regulating genetic elements in our bodies and in any organism's body. Um, and scientists are manipulating this to specifically target genes. One way that they're doing it is using toehold switches, which I will come back to. Bacteriophages have also been used as diagnostic tools. A bacteriophage basically presents no threat to humans as they only target bacterial cells, and they are used to ID pathogenic strains of bacteria. And the way they do this, it's called in vitro, and it is out of the cell. So basically, they take a sample from a host and they put it onto some media and they add the bacteriophages. It all takes place outside of the host body. Another biosensor approach is to use just bacterial cells as host cells for diagnostic indicators, and this one actually takes place inside the host body. So genetically modified bacterial cells are added into someone's body and they can ID both pathogens and pollutants. There was a test done in mice where they showed a very, very high diagnostic application to IDing a pollutant found in the mice's water. It was very effective. Coming back to the toehold switch on RNA-based biosensing. So before a toehold switch was developed, scientists had to rely on the RBS, the ribosomal binding site, which you see above that start code on there. That is the RBS, which basically tells the ribosome where to attach and start transcription of the mRNA. And this was a problem because scientists could only really target genes that came after these ribosomal binding sites. And once they figured out how to put a toehold switch on that RBS, that ribosomal binding site, they could really manipulate what genes they wanted to target after. They could kind of move around where that ribosomal binding site was in order to target new genes. The next major application of synthetic biology is microbial biosynthesis. So what microbial biosynthesis is exactly what it sounds like. It is using microorganisms to synthesize products. And the biggest target of microbial biosynthesis are plant natural products or PNPs. PMPs are used as flagrants, flavors, and medicines, and currently the harvesting of these products uses a lot of land, time, and resources. There is a lot of water, there's a lot of time and land that takes growing these plants. They have to grow through their full life cycle. They have to be cut and then broken down into these products. With microbes, scientists can streamline this process by manipulating the genome of microorganisms to synthesize these desired products. Now, there are a few requirements. One major requirement is the fact that there needs to be at least 4 enzymes involved in this process to produce many of these compounds, and these enzymes need to naturally occur in the host cells being used. Another drawback is plant cells cannot be used as they're not easily manipulated and their genomes are not well documented yet. So manipulating a plant's genome is just too costly. Using microbial biosynthesis, there are actually a lot of benefits. The biggest benefit being on-demand capabilities. When a chemical is needed or a product is needed, it can be used on demand. Um, It's also scalable and controlled, so the production of these chemicals can either be scaled up or scaled down depending on the needs of the scientists or the needs of the product. Um, and they have showed that these microbes can actually produce these products at a higher purity than many plants can. So that means that these products being created by these microbes are actually more pure and have to go through less breaking down to get the pure substance. Um, E. coli and Saccharomyces cerevisiae, are two microbes being used in this field a lot because their genomes are very well documented and it is very easy to manipulate their genomes. And this is just a figure showing that these microorganisms have the ability to produce all these different compounds that plants produce. All right, another major application is its application in the environment. Many microbes by nature can break down many compounds that other living organisms, such as humans, cannot use. Heavy metals are very, very toxic to most living organisms. However, there are bacteria that use heavy metals in their metabolism. Bacteria that can tolerate these conditions are known as extremophiles, and they are being manipulated by scientists to remove harmful compounds from the environment quickly and effectively. So currently, the main way that heavy metals are removed from the environment is through plants. Many plants are planted, they absorb the heavy metals into their cells through their roots. But the drawback with that is once those plants die and they decay, those heavy metals are reintroduced right back into the environment. In this case, if a bacteria is using a heavy metal in its metabolism, it means it's breaking it down into something different and it is changing its form so that after the bacteria leave, the heavy metals will no longer be present in the environment. And lastly, I would like to talk about both the ethical implications and the risks of synthetic biology. Ethically, there is a lot of pushback from the public, as many people think that scientists are trying to quote unquote play God by manipulating organisms in such a way. So there is a very ethical debate going on in this field on whether or not humans should be able to manipulate the genetic code of other organisms and kind of create new organisms. Um, and the biggest risk is the impacts on the environment. Introducing these bacteria into the environment, while the thought is good, and while the intentions are good, bacteria have the ability to share genes through horizontal gene transfer, which means that once a bacteria that has the genetically modified gene is in the environment, there's the potential that that bacteria shares that genetic information to the bacteria around it. Now, this can cause a very unknown effect in the environment, and the short-term and long-term effects are very unknown. If you have a group of thousands of bacteria with this gene, the risks and the results are very undocumented and they can be very severe. Um, Another major fear with synthetic biology is how it can be applied to manufacturing biological weapons. While the thought that all humans will use this technology for the good of our species is a good thought, there are people out there who tend to use these types of things in a negative way. Um, and synthetic biology poses a very, very big risk for biological terrorism. One example that I think of off the top of my head is there is a bacterium known as Bacillus anthracis, which actually produces the deadly compound anthrax. And if a scientist figured out how to hyperproduce anthrax in a lab and they really wanted to do something nasty with it, they could. And it is a very scary thought to have, as anthrax is not the only biological component that can be used as a weapon. Um, and yeah, there are many compounds that are produced by microbes that are harmful to humans, and this can be manipulated to harm people, which is a very, very scary thought. Um, so, in conclusion, synthetic biology has an extreme upside both practically and economically. In 2022, this field was actually generated $11.4 billion worth of wealth, and the field is only growing. In the next 5 to 10 years, there will be a lot of growth in this field, and I urge you to follow this closely, especially in the medical fields and the industrial fields. There aren't as many risks in the medical and industrial fields as there are in the environment. Um, Specifically, the medical field has the ability to grow a lot and the potential to push humans into the next chapter of healthcare and industry. Just like the Industrial Revolution was a major jump forward as a species for us, synthetic biology could potentially be that next big jump forward. Investing into bioremediation is a little more risky, as the adverse effects are higher. So if you do invest into the environmental field, just know the risks associated with it and be very careful. All right, that is all I have for you today. Thank you for listening to me, and I hope that my presentation helps you make a decision in the future.